morning students. Hope you all are fine and safe. Learn everyday's portion. Okay. In the previous class we had studied about origin of agriculture and organic agriculture. In organic agriculture we had studied about some kinds of biofertilizers such as rhizobium, azolla, arbuscular mycorrhizae, seaweed biofertilizer, etc. And also we had studied about aims of plant breeding. Today we are going to learn different kinds of conventional methods of plant breeding. They are plant introduction, selection, hybridization and heterosis methods.
Conventional plant breeding methods resulting in hybrid varieties had a tremendous impact on agricultural productivity over the last decades. It develops new plant varieties by the process of selection and seeks to achieve expression of genetic material which is already present within the species. Now we shall discuss about some of the conventional methods of plant breeding. First one is plant introduction. What in by plant introduction? Transference of genotype or a group of genotype of crop plants from the place of their cultivation into a new area where they were not being grown previously. Example, rice variety of IR8 introduced from Philippines and wheat varieties of Sonora 63 and Sonora 64 from Mexico. The newly introduced plant has to adapt itself to the new environment. This adjustment or adaptation of the introduced plant in the changed environment is called acclimatization. All the introductions must be free from presence of weeds, insects and disease causing organisms. This has to be carefully examined by the process called quarantine. That is a strict isolation imposed to prevent the spread of disease. Based on adaptation, plant introduction may be classified as primary introduction and secondary introduction. When the introduced variety is well adapted to the new environment without any alternation to the original genotype, that is called primary introduction. In secondary introduction, the introduced variety is subjected to selection to isolate a superior variety. Then the superior variety is hybridized with a local variety to transfer one or few characters to them. The botanical gardens in different parts of the world also played a significant role in plant introduction. Example, tea varieties collected from China and Northeast India, initially grown in Botanical Garden of Calcutta, from which appropriate clones have selected and introduced to different parts of India. Next one is selection. One major technique of plant breeding is selection. Selection is a choice of certain individuals from a mixed population for one or more desirable characters or traits. Selection is the oldest and basic method of plant breeding. There are two types of selection. One is natural selection and another one is artificial selection. Natural selection, that is based on a rule in nature and results in evolution. That is based on Darwin's principle, survival of the fittest. But this method takes longer time in bringing about desired variation. Next is artificial selection. What do you mean by artificial selection? It is a human involved process to select better crop from a mixed population where the individuals differ in character. There are three main types of artificial selection. They are mass selection, pure line selection and clonal selection. In mass selection, a large number of plants of similar phenotype or morphological characters are selected and their seeds are mixed together to constitute a new variety. The population obtained from the selected plants would be more uniform than the original population and are not individually tested. After repeated selection for about 5 to 6 years, selected seeds are multiplied and distributed to the farmers. The only disadvantage of mass selection is that it is difficult to distinguish the hereditary variation from environmental variation. Next is pure line selection. The word pure line was coined by Johansen in the year 1903. It is a collection of plants obtained as a result of repeated self-pollination. It is important. Repeated self-pollination from a single homozygous individual. Hence, a variety formed by this method show more homozygosity. That means similar characters we can obtain with respect to all genes. The only disadvantage of this method is new varieties cannot be obtained. Why? Because it is undergone repeated self-pollination. If it is undergone cross-pollination only, new varieties can be produced. Yes. Third type is clonal selection. Crops like sugarcane, potato, tea, banana and certain species of grasses are asexually propagated and produce very poor seeds based on their phenotypic, that is morphological appearance. The method of clonal selection is employed to select improved variety from a mixed population. Selected plants are multiplied through vegetative propagation to give rise to a clone. The genotype of a clone remains unchanged for a long period of time. Next one is hybridization. Hybridization is a method of plant breeding to produce new varieties by crossing of genetically unrelated parents. The first natural hybridization was observed by Cotton Mather in maize. Now, let's discuss steps involved in hybridization. First step is selection of parents. Male and female plants of desired characters are selected. Both should be tested for their homozygosity. What do you mean by homozygosity? Homozygosity is the state of possessing two identical forms of a particular gene, one inherited from each parent. Second step is emasculation. Emasculation is a process of removal of anthers to prevent self-pollination before dehiscence of anther. Then bagging. The stigma of a flower is protected against any undesirable pollen grains by covering it with a bag. After emasculation and bagging, crossing process takes place. Here, transfer of pollen grains from selected male flower to the stigma of the female emasculated flower. Can you name this process? It is called pollination. Yes. Last step is harvesting seeds and raising plants. The pollination leads to fertilization and finally seed formation takes place. The seeds are grown into new generation which are called hybrids. According to the relationship between plants, the hybridization is divided into 4 types. They are intravarietal hybridization, intervarietal hybridization, interspecific hybridization and intergeneric hybridization. What do you mean by intravarietal hybridization? The cross between the plants of same variety. Such crosses are useful only in self-pollinated crops. Intervarietal hybridization. Here, the cross between the plants belonging to two different varieties of same species. And it is also known as intraspecific hybridization. This technique has been the basis of improving self-pollinated as well as cross-pollinated crops. Third one is interspecific hybridization. The cross between the plants belonging to different species belonging to same genus. It is also called intragenic hybridization. Example, Gossypium varieties, that is cotton. Gossypium hirsutum and Gossypium arboreum. Here Gossypium is genus, is hirsutum and arboreum are different species. So this kind of hybridization is called interspecific or introgenic hybridization. It is commonly used for transferring the genes of disease, insect, pest and drought resistant from one species to another. Fourth type is intergeneric hybridization. The crosses are made between the plants belonging to two different genera. Example, triticale, first man-made cereal. This we had studied in chapter 3. Here the cross between Triticum durum and Secale cereal. The new variety is triticale. Here both parents Triticum and Secale are different genera. This kind of hybridization is called intergeneric hybridization. So, what are the four types of hybridization? Intravarietal hybridization, intervarietal hybridization, interspecific and intergeneric hybridization. Next is heterosis. Here hetero means different, cis means condition. G.H. Schull was the first scientist to use the term heterosis in 1912. What in be heterosis? The superiority of the F1 hybrid in performance over its parents is called heterosis. It is also called hybrid vigor. Vigor refers to increase in growth, yield, greater adaptability of resistance to disease, pest and drought. Vegetative propagation is the best suited measure for maintaining hybrid vigor since the desired characters are not lost and can persist over a period of time. Depending on the nature, origin, adaptability and reproducing ability, heterosis can be classified into two, euheterosis and pseudoheterosis. Euheterosis, here U means true. This is the true heterosis which is inherited and is further classified as mutational euheterosis and balanced euheterosis. Mutational euheterosis is the simplest type of euheterosis and results from elimination of unfavorable trait or character by their superior dominant alleles in cross-pollinated crops. Balanced euheterosis is well-balanced gene combination which is more adaptive to environmental conditions and agricultural usefulness. Pseudoheterosis. Pseudo means false. This false heterosis, it is also termed as luxuriance. Here the progeny possesses superiority over parents in vegetative growth but not in yield and adaptation. This pseudoheterosis plants are usually sterile or poorly fertile, which means such types of plants are not involved or poorly involved in reproductive process. Now let's conclude the session. So far we had discussed plant introduction and its types, selection, its types, hybridization, its types and steps involved in hybridization, and heterosis and its types. Thank you, students.